Okay, hello friends. I'll begin recording the video for what we can say, that is our next portion, or allied portions of our. Okay, so I'm ready. Yeah, allied portions of our sixth chapter, isn't it? That we are doing. Molecular basis of your chapter's name is molecular basis of inheritance, and I have already begun this chapter with certain basics that I have provided, and you have well understood about the structure of DNA and other things, introductory things that I have given. I have also explained you, so DNA structure is already explained in the previous video, and central dogma is explained. Okay, concept of gene and central dogma is explained, that how genes function and how the DNA is the genetic codes the genes and how the characters develop. All these things were discussed, and how it occurs and all. Yeah, so today basically we have understood the covalent structure of DNA. In our previous video we have understood the covalent structure of the DNA in the previous video, isn't it? That is the you can say the secondary structure. Okay, because the double helix, if I think that this is the nucleotide and nucleotide are doing phosphodiester bond, this is the secondary structure, primary structure, and there are two strands which are complementary to each other, A bonding to T and G triple bonding to C in this way, and the formation of the double helix or existing in double helix structure, we can call it a secondary structure. So this becomes the primary, this becomes the secondary structure. And today we will learn basically about the tertiary structure of DNA. Basically we will learn about the tertiary structure of DNA. I hope that you are well accustomed with it, understanding tertiary structure. Why I am saying you are well accustomed? Because you have known since class 9, you have heard this name chromatin. Okay, so basically we are going to talk about chromatin only today. Now before going into any details, first of all I want to ask you a question. If there are base pairs in our DNA and one base pair, the distance between the two base pair, that is one base pair distance itself from the other base pair with 34 Armstrong, 0.34 nanometer, so 3.4 Armstrong. So I think 3.4 Armstrong, so which gives me 0.34 nanometer. Now if there is 0.34 nanometer distance, if I convert it into meter, it will be, I think, 0.34 into 10 to the power minus 9 meter. So that much is the distance between the two what we can say base pairs. Now Human Genome Project HGP revealed that there are almost 6.6 into 10 to the power 9 base pairs in a human DNA. So if you multiply this number with this, you will get the entire length of the DNA, is it or not? So when you multiply this number, you almost get 2.2 meters. So this 2.2 meters means a length of a normal table, a length of a normal dining table. Wow, a length of a normal dining table. So you can imagine the number of cells that we have, and if we add up if we take out the DNA of all the cells and place them side by side, I think it will go up to the moon. Always, very popularly I say it, yes or no? So those who are my old students, they know. However, my point being that such a huge length, how can it fit in a nucleus which is even few micrometers in diameter? Is it or not? So definitely the answer is that there is supercoiling of the DNA. Supercoiling of the DNA. So the DNA coils itself. So why do I mention the term supercoiling? Because DNA is already a coiled structure, forming the double helix in this way. Now, coiling of a coil is called a supercoil. So that's why I am saying supercoiling. Telephone wires of old days were like this, land phones, land telephones. Land telephone wires of olden days were like this, and they used to get coiled among themselves in this way. And you must have, if you have not noticed, I don't think that you have noticed, we did notice in our childhood times that land telephones had this kind of wire and they used to get coiled among themselves. So it used to form a supercoil, and it's a commonplace notice that they used to occupy smaller space at that time when they used to coil. So definitely the DNA, when it will coil up or supercoil up, then it will occupy very less space. That is very, very true. Yes or no? So now, yeah, definitely this supercoiling does occur. Now this supercoiled DNA, how they coil, there are various ways of coiling. There is positive coiling, there is negative coiling, coiling from this side or from that side, like there is right-handed and left-handed DNA. In the same way, coiling is there. But details of supercoiling is much more higher class, so I will not go into the details of supercoiling. I just wanted to tell you that there is supercoiling of the DNA, which is the solution of the problem that such a huge DNA can be fit inside the small nucleus. Now, moving ahead, you see what happens is the DNA in its supercoiled form, what it does is that it winds up, that means पेचिए जाए around positively charged histone proteins in case of eukaryotes. That is the truth. Positively charged histone proteins. Sir, why the histone proteins are positively charged? Histone proteins are positively charged because they contain huge amount of lysine and arginine. That is the reason why histone proteins are positively charged. Now, why do we need a positively charged protein for coiling of the DNA? Then you must remember that DNA has phosphate group, yes or no, which is negative or negatively charged. So DNA itself, I can say that DNA is negatively charged. Now, as because DNA is negatively charged, what happens is it gets itself comfortable to coil above the positively charged protein. So I hope that up to this much you have understood. Now, the fact is this, that okay. So the fact is this, I am having a little bit of net problem. I think later on there will be more net problems, so I am recording the video now only. Otherwise, I really wish to teach live today, but I was fearing the net problem. Anyway, so you should remember that histone protein is an octamer. What is the meaning of octamer? Octamer means that there are 8 units. That there are 8 units of protein linked with each other, 4 like this and 4 at the back. Okay, just back, at the back 4. So in this way histone I am very poor in drawing, I'll try to draw 4 like this, okay, and 4 at the back. I'm just showing certain portions at the back. Okay, so it's an octamer and the DNA wraps around the octamer. Do you know that how much length of the DNA does actually wrap around one histone octamer? 200 base pairs of DNA. 200 base pairs of DNA. So that means only 200 base pairs of DNA wraps, and then it comes, and then again there is another, suppose this is octamer, 200 base pair wrapped around, then it again comes and again wraps around, another 200 base pair wraps around another octamer, again it comes and another base pair wraps around another octamer, and in this way it goes. Each of these is known as nucleosome, and the entire model is known as the nucleosome model. So that is the reason when now this entire chromatin thread. Now if you have understood up to this much, I would like to mention that in the nucleus, the entire chromatin thread that you see, okay, is actually repetition of the entire chromatin, is actually the repetition of nucleosomes. That is so obvious. Yes or no? So I hope that you have understood. Now, time for some questions. How many nucleosomes do you think would be present in the human cell or human nucleus? Definitely, if it is 200 base pair of DNA that is wrapping around, so 200 base pair into 0.34 into 10 to the power minus 9 meter, this much length of DNA. So this is the total length of DNA which is wrap around one nucleosome, and the total length of the DNA is 2.2 meters. So if n is the number of nucleosome, so n into 200 into 0.34 into 10 to the power minus 9 must be approximately equals to 2.2, from where you can calculate n. So that is the funda. So I hope that the question that I have asked, that is clear. Okay. Remember that under the microscope, the entire chromatin appears as beads and threads structure, or beads on string structure. So that means there is a string with bead, string. So there is a string with lots of beads on it. Like for example, if you take a thread and if you make a knot here, then make a knot here, then make a knot here, make a knot, automatically the entire thread will become very small with lot of knots. Yes or no? So that makes sense. The entire DNA is becoming much more smaller. Our entire chromatin structure is also becoming much more smaller because of the presence of knots. What are these knots or beads? these are definitely the nucleosomes. I hope that you understand. So this was observed under electron microscope. Remember, this was observed under electron microscope. Under the electron microscope it was observed. If I show you in your NCERT book itself, see, bead-like structure, bead-like structure, bead-like structure. So that is what I was saying. So that is what, Now talking about the chromatin, the structure that you have just understood becomes chromosome, which I have explained earlier also, during the cell division or metaphase stage of the cell division that you have learnt earlier. Entire chromatin breaks down and becomes much more condensed, becomes much more condensed. Okay. Now, conversion of the chromatin to chromosome, if you convert the chromatin to chromosome during metaphase, it does requires the assistance of extra proteins, and those proteins are known as NHC proteins. Now, what is the full form of NHC proteins, which converts the chromatin into chromosome, or assistance of these proteins helps the chromatin to condense even more further, Okay, so these are non-histone chromosomal protein. Non-histone chromosomal protein. So yeah, that is it. Now you will see that it was found that in some portion, in a chromatin, some portion or some beads were loosely packed, and they were known as euchromatin, and some were very prominently and highly packed, and that portion was known as heterochromatin. And that portion was known as the heterochromatin. So, I hope that you understand this. Now you will be raising question in your mind, Lord, how did they understand? Because under the microscope, when a typical staining is done, this portion stained very light. And because this portion stained very dark. Now it is very obvious that the accumulation of the NHC protein, the histone proteins, and the coiling up is much more loose. That's why it is staining light. And here the coiling is much more tough and high. That's why it is much more condensed, and the color of the stain is entering and getting trapped even more. More amount of trapping is there of the color or the stain. That's why it is staining dark. Suppose, for example, if you take a thread and create a very light knot, and here you create a very dark heavy knot, then you allow color to flow, soak. Okay, color is soaking. You are allowing color to soak through capillary action. After some time you will show that it has soaked more color, because more color is trapped here. So that means this is much more highly packed. So in this way they knew that okay, so that is more highly packed. Okay, so yeah, that is the thing. Now, question arises, why so? Why so? What was the requirement of this heterochromatin and euchromatin portion? See, euchromatin portion those portion of the gene or the DNA, sorry, or those portion of the DNA where the coding regions are there. Where there are coding regions. So yeah, there are this what we can say, yeah, okay. So euchromatin portion consists of those portion of the DNA where there are coding regions. Coding region means those regions of the DNA, those regions of the DNA from where the protein will come out, or maybe the messenger RNA will come out. If you have seen my previous video, you will know it. And the messenger RNA will code for the protein, which you have learnt in central dogma. So that is known as the coding region of the DNA. So coding regions are basically found in euchromatin, and non-coding region are packed in the heterochromatin. So it is as easy as a concept. Now, because when you have to go through the, or when the cell has to go through the central dogma, when the cell has to go through the central dogma, that is, the DNA will give rise to, or the DNA's coding region will give rise to a messenger RNA, and hence the messenger RNA will give rise to a protein. Then the supercoil of the DNA has to be opened, and the ATGCC, TTGC, the sequence that is present in the DNA has to be exposed. So that means the supercoiling has to be opened. So I hope you can understand that it won't be so fruitful to keep the coding region in the heterochromatin. That's why in euchromatin there is less coiling, so that it is opened up on and on. I hope that you have understood this. Now remember that heterochromatin are again of two types. Heterochromatin are again of two types: the facultative heterochromatin and the constitutive heterochromatin. Now what happens is, in case of the facultative heterochromatin and the constitutive heterochromatin, what I want to say is that it's this type of heterochromatin always will contain non-coding region and never code. Like for example, the centromere of the chromosome is formed of constitutive heterochromatin. In the chromatin thread, wherever there was the constitutive heterochromatin which never codes, that goes into the centromere. And you can understand why. Because these genes are not being expressed, so the expressive genes should be here. And facultative heterochromatin are like the present in the X chromosome or one X chromosome of females, where it becomes, inactive when it goes into the polar bodies. Isn't it? So if you have understood previous chapters, previous video, you will understand this, what I have said here. One of the X chromosomes, the facultative heterochromatin is present. Not throughout the chromosome, but maximum of the X chromosome is formed of the, that is facultative heterochromatin, okay, which goes into the where? The polar bodies, which is not required. So basically the genes are not lost. The genes are not lost. You think that going into the polar body of the, when oogenesis occurs, the genes are lost. So basically the genes are not that much lost. A lot of heterochromatin goes into the inactive, it renders it inactive, renders this X chromosome inactive. So I hope that you have understood. If your biology concept is okay with my previous video, then you will understand what I have said here. And another thing that I will mention is that, remember, the heterochromatin has a tendency to extend and repress. Repress means suppress the gene expression. Gene expression means producing messenger RNA. Okay? Gene expression, to extend and repress the gene expression of the euchromatin portion, which is what I want to mean. That suppose, for example, this is the thing, and suppose the beads and all these things are there. Suppose this is the heterochromatin portion, this is the euchromatin portion. It tries to extend, and when the heterochromatin will extend and more supercoiling will be there, the important genes that were there, they will be repressed. So it can extend and repress the important genes. So the boundary maintenance between euchromatin and heterochromatin is very, very necessary. Is very, very necessary. And it is done by the histone proteins only, or modification by the, it is done by the modifications of histone protein. How? The heterochromatin and the euchromatin, does contain different histones, and this difference is created by the modification of the histone, little bit structural change of the histone, different change in heterochromatin, different change in euchromatin, and that way the boundary is maintained. So I hope that you have understood the eukaryotic thing entirely. Now going into the prokaryotic thing. See, prokaryotes do not have a nucleus, right? Prokaryotes do not have a nucleus. In their cell, the DNA is supercoiled around the protein, which is called histone-like protein. But basically it is also known as HU protein. The HU protein has two subunits, HU alpha and HU beta. These two types of proteins are situated, and they are positively charged, as because DNA is negatively charged, and they coil around and stay in the cytoplasm only, as the middle of the cytoplasm. So that is the prokaryotic thing, you should remember. Okay, and that entire structure is known as nucleoid, not a nucleus. That entire structure is known as the nucleoid, and not the nucleus. So that is what I wanted to tell you. So I hope you have enjoyed the supercoiling of, what we can say, or packaging of DNA into the cell. So yeah, that much is it, and then I will move into my next topic. I am sending you these notes.